What happens when we fast? The main functions of the body organs is to mobilize energy resources for various tissues to use. The two important energy resources that are needed are glucose and fatty acids. Glucose is mobilized from the liver's glycogen stores and released into the bloodstream through a process called glycogenolysis. Fatty acids are mobilized from adipose tissue, which stores fat in the form of triglyceride or triacylglycerol and releases it to the bloodstream during fasting. The glucose output from the liver is a consequence of the rise in epinephrine, glucagon, and cortisol in the plasma. These three hormones, individually or collectively, will all activate glycogenolysis and gluconeogenesis, the two processes that can restore glucose to the bloodstream. When glycogen stores get depleted with time, gluconeogenesis becomes a more important source for glucose output from the liver. Gluconeogenesis will typically use the carbon skeleton derived from glucogenic amino acids. During fasting, for the process of gluconeogenesis, the liver picks up amino acids from the bloodstream to produce the corresponding carbon skeletons that are needed in gluconeogenesis. In turn, these amino acids that are in circulation are typically released from breakdown of normal muscle proteins during exercise. So during fasting, there's an increase in amino acid derived from muscle proteins in circulation. This will provide the liver with the glucogenic carbon skeletons needed for gluconeogenesis. During fasting, the second important energy source needed in the bloodstream are fatty acids. Fatty acids will be derived from stored triacylglycerols in adipose tissue. This is accomplished through the activation of hormone-sensitive lipase, which is enriched in adipose tissue and is activated by epinephrine. Hormone-sensitive lipase gets phosphorylated by protein kinase A, which is in turn is activated by activation of the beta adrenergic receptor by epinephrine. This results in the release of 3 fatty acids from triacylglycerol and glycerol to the bloodstream. During fasting, fatty acids released from adipose tissue by the actions of hormone-sensitive lipase will be released into the bloodstream and transported by binding to serum albumin, which allows many tissues to pick up the fatty acids and use them as an energy source. Some of the important tissues that can use fatty acids are the liver, heart, skeletal muscle, but not the brain or red blood cells. Once picked up, fatty acids are metabolized primarily through beta oxidation in the mitochondrial matrix to provide energy for these tissues. During fasting, the liver switches to using beta oxidation of fatty acids as an alternative energy source. When its needs of energy are met, acetyl-CoA in the liver begins to accumulate. That acetyl-CoA is then shunted towards the synthesis of ketone bodies, acetoacetate and beta-hydroxybutyrate. The importance of ketone body synthesis in the liver is that it provides a new energy source that is easily transportable in the bloodstream, so it can be picked up by other tissues, whereas acetyl-CoA is not. Ketone bodies can be used by many tissues, including the heart, brain, and skeletal muscle, but not the liver.
If you like this video, please subscribe. Thank you.